So, the other thing administration has done, that they've done for the past couple of semesters, which is why I had kind of set the deadlines where I had set them before, for the case study. So I'm going to go ahead and, actually I thought I had opened it, but I think I did not. Open a gazillion other files, right? That's any good for people who can't seem to make it here. They make it on test day though. You got to take the test Friday, right? If for some reason something happens to you, still come talk to me, call me, text me, something, right? Don't just take a 0 on the test, but you are not going to take the same test as everyone else if you do not make it here by Friday. Does that make sense to everybody? Because I'm not going to hold everyone else up from being able to see their test results to help them study for the final because something happened to you. Okay?
You want me to remind you about case studies?
Mm-hmm. Yep, thank you. So as it relates to case studies, as I said, right, because grades, I don't have until the last day to turn in the grades for your guys' class because your test, your final exam is Tuesday. I do have a couple of days, right? So I think I moved the deadline. I'll have to look for sure. I changed it to May 5th, right, to give you guys some reprieve. So you can really concentrate on test 3, right, that we have this Thursday, or get your ass here Friday, right? Otherwise, you're not taking the same test as everyone else, right? Because I'm going to make those results available Friday afternoon so everyone can review over the weekend and Monday in time for the final on Tuesday, right? And then, as I said, I'm going to make an announcement probably later today regarding how we're going to do the final, right? Whether we're going to be stuck doing Scantron or we can sneak in after the other class in the computer lab that Tuesday. And then make sure that you complete the case study exam and the case study assignment before May 5th at midnight, because I'm going to be calculating your grades on that Friday to get them in by the deadline. Because otherwise I'll get put on the naughty list, and I don't like the naughty list. So, all right. And again, what do you do if you run into difficulties? You communicate with me. I'm not chasing after you. You come to me. Okay? I'm not going to have time to chase. There'll be one last assignment posted this week. It's the easiest one you've probably done all semester long. It's a survey, right? At the end of the semester, I always give my students a survey, and I value your input so much that I give you points for completing it. Even though it's anonymous, it puts a little check mark that you've completed the survey in the grade book, and I will give you your points. It'll give you your 15 points or however many points I have written for it in the syllabus. I don't remember offhand. You got to look at the syllabus. I tweak those questions, right, each semester, and I really do listen to your guys' feedback. So sometimes I try different things, right, and I ask for your guys' feedback on the different things that I've tried. Sometimes I ask you guys, I'm thinking about trying this. What do you think about it, right? And that really helps me, you know, see the pros and cons of things that I think may be great, but my students are like, oh, no, that would suck for me, right? So I really do read and take into consideration feedback from you guys, and I really do appreciate it. So please take the time to answer those questions with some thought, which is why I do it as a survey on Canvas so that you can do it at your leisure.
Where are the modules as a survey?
It's not there yet. I'll probably put it under the syllabus and stuff module, but I'll put a link and an announcement like I always do for things like that. So also don't forget to finish up your Connect assignments, right? And you can redo any of those, right up until Friday when I download grades, and that's it. You know, once I start finalizing grades on Friday, that's it. If something happens to you, though, what do? You come talk to me, right? Because the absolute latest I can make a change to a grade is Wednesday at 1 o'clock. That's when grading shuts down. So you'll notice final grades due 1 p.m. Wednesday, the 11th, right? And again, this is for Monday and Tuesday exams. The Monday, and there really is only one exam on Tuesday. No one's taking 7 o'clock in the morning classes in this class for sure. So that's it. I mean, that's when I gotta, that's the absolute final deadline, that any changes can be easily made for the semester. So as I said, this is my schedule for final exams, and when my office hours are, and then later the following week, it's, just contact me. I'll be around, right? I don't evaporate at the end of the semester. I'll unfortunately still be working. I don't get vacation until August 1st. So, any questions on any of that? Okay. So, the other thing that I noticed that I neglected was there is an objective for you guys that I don't cover in my other class that is on your test. So, I posted a separate PowerPoint, you'll notice, last night under this module. And it's just a few slides. It's Disorders Part A. It's the first thing I'm going to go over today. And then, as I mentioned earlier, and I was handing it out, and if you came in late, I hope you picked one up, but I also posted it in 2 places. I posted it here, and then also in the main module. Your book, your current book, which thankfully we voted to get rid of, starting in the fall. Students won't have the horrible book you have. They don't do a good job of covering type 3 hypersensitivity. So this is from the other book, from the other class, on type 3 hypersensitivities. So I printed up some for you guys so you could have it in class to look at, but also I posted it online for you guys. And this is it, y'all. It's the last lecture. That's all she wrote. So, don't forget to do your Connect assignments. Don't forget. I think that's it. Okay. I don't think of anything else I've forgotten. If I did, oh well. Right? So, we want the Part A. So this objective I can guarantee you is on the test. Right? So make sure you go over and understand these definitions that we're going to go over right now. So this is probably not on your objective sheet you printed out previously. For each one of these, you need to be able to identify an example. So typically what I usually do is I give you an example, right? Examples are usually better for people to understand than just definitions, right? I prefer definitions. So imagine a matching or multiple choice where you're given an example of these, and if you know the definitions, definitely you should be able to match up the example with the term. So this is a really good picture that shows the 4 different principles of immunity. So immunity can be natural, right, or it can be artificial. It can be active, or it can be passive. So when we define them as whether they're natural or artificial, active or passive immunity. So these are some excellent examples of them. So you'll see for natural active immunity, this is when immunity results from the individual when they're exposed to an infectious agent, right? So for me, I have natural active immunity to chickenpox. I had the chickenpox. It was brutal. I was 4. I still remember it, right? But now I have antibodies against, right? I have immunological memory against the chickenpox virus. My son, on the other hand, right, has hopefully an active immunity to chickenpox. But he did not get this naturally. He got it artificially because the doctor gave him a vaccine, right? Whenever we stick you with a needle, that's artificial, right? That isn't natural, right, to get stuck with a needle, right? So vaccination is an example of active artificial immunity, right? Artificial in that we're purposely giving you, right? You weren't naturally exposed to it. We're purposely giving you the pathogen to get your immune system to actively respond, right? So he has memory cells, right, hopefully that recognize this virus and antibodies that recognize it. So he'll never, hopefully, ever actually get chickenpox. Even if he's exposed, right, his immune system's already seen it, is going to deal with it. I also gave him passive immunity against things that I was naturally or artificially exposed to in my lifetime because I breastfed my son. That is a natural form of immunity that you can pass on to your children via the breast milk. You continually do it in the breast milk in the form of a particular class of antibody. What antibody is always secreted in your secretions and is also secreted in breast milk? IgA, right? So, but did the baby make that IgA? No. So does that baby have memory cells on how to make that IgA? No, right? This is passive immunity. That's limited, right? Because you're passing it. Active has memory. Passive does not. That's the important distinction between these two types of immunity. But passive is great, right? It can be very protective. And then in the colostrum, what class of antibody do we pass on to our baby? IgG, right? Which is going to help protect them in the first couple months before they get vaccinated, right? Before they're exposed to things and develop their own active, natural, or artificial immunity. And then the other one that most people probably will never get in their lifetime is artificial passive immunity, where we give it to you from someone else artificially, right? You're not sucking on your mom's boob to give, right? Someone is sticking you with a needle and giving you someone else's antibodies. Typically, that's what we're giving you, right? When we talk about these type of immunities, especially with passive, we're talking about antibodies. So again, when we give those antibodies to that person, can they protect them? Sure, otherwise why would we give it to them, right? Kind of a useless shot, right? We're giving them protection. But did they make it? No. So once those antibodies are gone, they're no longer protected. They got to be exposed themselves and make those antibodies. So why would we do that, though? Why would we give somebody antibodies? Why not just vaccinate them?
Not all vaccines work.
Not all vaccines work. Can all people get vaccinated? No, not all people can get vaccinated. And typically, how long does it take before your body makes an antibody? 7 to 14 days on average. If you have tetanus, do you have 7 to 14 days to wait for your immune system? No. Right? And you have the infection, right? Which means it's already too late. Vaccines are what? Prevention. Prevention. Right? Which means you've got to take it before you have the infection. If you have the infection, we've got to treat. Right? So in that case, we've got to give you antibodies someone else already made. So with tetanus, if you have tetanus, you haven't been vaccinated recently, your titer's low, you don't have a lot of antibodies, they will give you someone else's antibodies to help you.
What about medical steroid shots?
That's totally different. That's not going to infer any type of immunity. Remember, we're talking about immunity here.
I just want to make sure it was one of the two pretty official.
Doesn't classify at all. If you get a steroid shot, that's an artificial way of being exposed to something. But remember, we're talking about immunity here. Steroids aren't going to give you immunity. Does that make sense?
Okay, I just want to make sure.
Yeah, we're classifying immunity here. Does that make sense to you guys? All right, so for me, I think the examples really help in the understanding, right? But straight up definitions, right? Naturally acquired immunity, right, means that your adaptive immunity has been triggered, right? You get memory cells, you get antibodies through natural events, right? Being exposed to a pathogen. Immunization, of course, mimics these events, but we're artificially giving it to you. You're not naturally exposed to it, we're artificially. We're sticking you with a needle or shoving something up your nose or whatnot, right? But you're still going to actively respond, right? And that's the benefit of it. You get those memory cells, right? You get those antibodies before you see the really bad guy. So remember, we classify things as natural or artificial, and then is it going to be active, right? Is your immune system going to respond? Are you going to produce memory cells and antibodies? Or is this something that's been passed to you? So passive, we said, occurs naturally through pregnancy, right? IgGs cross the placenta. It's another way that we protect. And then IgA, all the time in the breast milk, but also IgGs pass in the colostrum, that first milk that the mom produces. Artificial passive, right? If we pass something artificially, it was produced by another person or animal, right? And we can use this to prevent disease before or after likely exposure. And this is typically referred to as anti-serum because, as I said, typically we're giving antibodies that were in someone else's blood, that liquid phase of your blood that's referred to as serum. So we can help prevent hemolytic disease of the newborn by giving the mom antibodies against the Rh antigens that she doesn't have because, right, she doesn't have it, so she's not going to make those antibodies. If we give them those antibodies, if we give her RhoGAM, any red blood cells that get into her system that are positive, that don't match hers, those antibodies will take care of it. But the good news is, right, that she's not making antibodies that are passing to the baby who's positive. Right? Like I said, with tetanus, we can give somebody antibodies that someone else produced and protect them. If you get bit by a snake, right, and that venom, right, that toxin is in your system, we can give you antibodies against it, right, very quickly, right? Because you don't have time when it comes to those serious toxins for your body to make antibodies. We got to give you someone or something else's antibodies. So we briefly started talking about these different types of hypersensitivities, right? The 4 different hypersensitivities. Remember I said I would refer to them by their full names. I wouldn't just say type 1, 2, or 3 on the test. I'll write them out. And then I don't think we're going to get to autoimmune disease or immunodeficiency today, but read over that, right? There's not a lot of questions, and I think you guys can handle it. Not worried about that as I am the hypersensitivities. So for each one of the hypersensitivities, again, I want you guys to be able to recognize examples of these hypersensitivities, and especially for type 1, immediate IgE mediated, there's 4 different examples of things that could go wrong or could happen with this type of reaction. So there's a cool animation that you can watch, right, to help you in the understanding of this. These types of allergies, remember how our immune system functions, how our body functions, all this information is encoded in our DNA, right? So if your system has a tendency to go the IgE route. That's encoded in your DNA. So if you happen to be in my family, right, my DNA I got from my mom and my dad. My mom has allergies, right? So do I. So does, unfortunately, my son, because we were all genetically linked, right? So you hear more and more about peanut allergies nowadays, right? The reason for this is, again, it's genetically inherited. But why is it more common now than it was before? One very simple reason. Survival of the fittest does not work on the human population, right? We prevent the death of individuals in our population with medical treatment and the knowledge we have about how things work. What would happen to people previously who had peanut allergies? Severe peanut allergy, I should say. They died, and probably when they were a kid, because we love peanut butter in the United States, right? So you died as a kid because you had a severe peanut allergy. So you didn't grow up to be an adult, did you? You didn't have children, did you? No. You were eliminated because you were not fit, right? Natural selection worked on you. Natural selection does not work on the human population for the most part anymore, right? Or the one animal population not affected by survival of the fittest. So we have these problems, you could say, increasing in number because we don't have that selection happening to the human population as it relates to certain things that we understand what is going wrong now, and we can avoid that happening. Kind of crazy, huh? When you think about it. Right. I sometimes joke, if natural selection had happened, I wouldn't have made it. Right? I'm probably not I have not been the fittest in my lifetime. But that explains why we see more and more of this, right? It is our genes, right? And us continuing to perpetuate these diseases or these abnormalities, you could say, or predispositions. So type 1 reactions can be classified as local anaphylaxis, right, where it's happening in a local area. So people like myself who experience what's referred to as hay fever, right, where you inhale allergens, it affects your eyes, your nose, right? That's local. When you ingest something that goes into your bloodstream, like peanuts, for some people, that can be a systemic, a generalized, system-wide exposure. And that's when it becomes life-threatening, right? When something is affecting more than a small area, right, it's affecting a large area of the body. So when anaphylaxis, right, that's a term given to IgE-mediated allergic reactions, right? And when it's systemic, right, it typically causes an individual to go into what's referred to as shock, right, where you have a severe drop in blood pressure because you have widespread inflammation in response to this reaction. So the very first time you were exposed to that peanut butter, probably nothing happened that you were aware of. But behind the scenes, you just became sensitized because what happened is your immune system, you have B cells that recognized that allergen, right? And it's, instead of calling it an antigen, we call it an allergen because of the type of response. But in truth, it's an antigen, right? It's something foreign to your body that your body is recognizing. A helper T cell came along and convinced that B cell, when you make and secrete that antibody, you're going to make IgE. Because I think that's what's best for this bad guy that came in that you're seeing, that I'm also recognizing. Because remember, they're communicating with each other. And this is MHC class what that the B cell is presenting that antigen via? MHC class 2, right? And this is a helper T cell, so that's CD4, right? And he's recognizing, he's communicating, he's releasing cytokines. So this B cell becomes activated, right? It proliferates. He produces some memory cells, right? So when you get exposed to this peanut butter, this is all going to happen again, where you produce even more plasma cells that are secreting what class of antibody? This is the problem, is this class, right? Because what IgE does is it binds to Fc receptors, right, for that stem portion of our antibodies that are IgE antibodies attached to mast cells and basophils. So that when you, the second time, whether this is an hour later, right, or 3 days later, or weeks later, these antibodies will stay attached to these basophils and mast cells for weeks. So once you've become sensitized, once you've produced these IgEs, the next time you're exposed, your reaction is immediate, right? Literally can happen within seconds. When you inhale that pollen, it binds to these antibodies on the surface of the basophil or mast cell, and it immediately degranulates. It releases substances. One of them we're most familiar with people who experience allergy. What's one of the major mediators of the reaction? Histamine. This is why a lot of individuals like myself take antihistamines. What do these do? These compete with the histamine for the histamine receptors. When you take antihistamine, right, it binds to the receptors and it doesn't cause the receptors to activate. We don't get that inflammatory response. So that way, right, all those receptors are taking up with the antihistamine, but of course this doesn't last forever, right? This is why these drugs you have to take once a day, right? Which is pretty good, you know. Could be worse. You could have to take it like every 12 hours or something, right? But when do you take an antihistamine? Before you're exposed. Because again, it's got to bind to those receptors so that when the histamine's released, nowhere for the histamine to go. No receptors available for that histamine. So some people are like, I took my allergy medicine, but it isn't working. Well, that's because you went to your friend's house who has a cat that you're allergic to, right? And this reaction's already happened, and then you're taking the medicine. Doesn't work that way. You take the medicine, then you go to your friend's house, right? When you take it after, you're stopping any other reactions from happening when you're exposed again. You can't do anything for that exposure that already happened. You can't prevent it. You have to wait for your system to clear it out. But the good news is it's not going to happen again if you have antihistamine in the system. So people say, oh, my allergy medicine doesn't work. It's like, no, you don't understand how allergy medicine works. It's a preventative. Now, some of them contain things like decongestants. Yeah, that's to deal with after the inflammation has already happened to get the fluid out of the area, right? But you're supposed to take it before exposure, right? So every morning before me and my son leave the house, we take our allergy medicine, right? This morning he's like, Mommy, my nose is stuffy. I'm like, don't worry, I'm getting it, right? We do our Flonase. He does his Claritin. I do my Allegra, and we go on our way, right? We breathe easy for the rest of the day. But I mean, you know, in the whole big scheme of things, right, I'll take that over peanut allergy, right? Because what do you have to carry around with you in case you accidentally eat something you're allergic to?
EpiPen?
An EpiPen. What does that contain? Epinephrine. That's a Band-Aid, y'all. That's all it is a Band-Aid for you to get to the hospital, right? Because what happens with that, again, widespread inflammation, dilation of blood vessels, blood is not getting where it needs to go. Epinephrine speeds up heart rate, constricts blood vessels, right? So it helps get the blood where it needs to go to those vital organs, right? But that's it's a Band-Aid. It's a temporary fix. Now, quite honestly, I personally do not, unfortunately, very fortunately, do not have that type of allergy. So I don't know, and I haven't investigated what they do for you when you get to the hospital. But I think it depends on what the allergy is and to the extreme. But But don't think that those people, they're like, oh, they could just, EpiPen themselves and they're going to be fine. No, that's a Band-Aid. It's a Band-Aid, right? May not even be enough. And this is why, parents freak out about this type of stuff, because their kid could die, right, from something they ingest. Now, some people think they're allergic to things like, my mom's friend told her she used to buy a new toaster when I come and visit in August because I can't have gluten, right? No, I'm gluten intolerant, which means my body does not tolerate it well. I'm not allergic to it. I'm not going to have an allergic reaction. Minute, small amounts that may be in her toaster, if she is kind enough to buy me gluten-free waffles, I can still eat and use her toaster, right? It's people with like celiac disease and stuff that have true, that's not even an allergy per se, but, have severe reactions. So a lot of times people think that they have an allergy, but it's really just an intolerance. If you have an allergy, it's pretty severe. It's pretty severe. And again, each time you're exposed, you go through this process. So you start producing even more IgEs and even more memory cells to produce even more IgEs, and it's just this vicious cycle that's never going to end, right? Which is why we want to try and limit exposure. Which is why, treatment-wise, you guys are probably going to think I'm slightly crazy for a second, right? So individuals can have hay fever like I expressed, right? They can have systemic, right, where they ingest stuff. They can also have where a reaction happens on their skin, commonly referred to as hives, or the wheal and flare reaction, where you see the redness, right, and the puffiness, the wheal. This is actually a skin test to determine what somebody is allergic to. I went through this many years ago. It was horrible. My arm swelled up so bad on this side where I wear my watch. I couldn't wear my watch for weeks and had to take steroids to get the inflammation down from the testing. It was that bad against thistle. And ragweed's pretty darn bad too. And a few other grasses I'm not too keen with. They're not my best friends. All the animal danders and stuff they did on my other arm, I wanted to be a veterinarian at that time, so I threatened to cut my arm off if it started to puff up, because I desperately wanted to take care of animals. And I have animals, other than, the two-legged kind. I get the four-legged kind too. But thankfully I'm not allergic to any of the things that are associated with them. A lot of people are, though, unfortunately. And I didn't at that time have any food allergies, and even to now, like I said, I just have an intolerance, not necessarily an allergy. There are lots of foods out there for people like myself who are allergic, individuals that are said to be, that have been scientifically proven to be inflammatory, and gluten is one of them. A lot of the grains, if you're experiencing a lot of inflammation, cut them out. You'll feel a whole lot better. They don't help with inflammation. Then you have people who are asthmatics, right, where their reaction is happening in their lungs, right, and that can be life-threatening. And they take different types of drugs. It's a different type of reaction. Histamine isn't one of the major players in your lungs. It's other mediators that are the major problem. So some people will get hives too when they ingest something, right? And this is, again, because when you eat something, where does it go? Into your blood. And your blood feeds your whole body. So eventually that allergen also gets to your skin, and you can have a reaction in your skin. When it's a food that is causing your hives, where do you usually see the hives first? Your stomach. Yeah, and I guess maybe your mouth, but I mean most people it's their stomach and then it spreads up their torso. Because again, think about what part of the skin gets the blood first. The one closest to the abdomen where the food is absorbed, right? They're the first to get that contaminated blood, you could say, right? So you can get this from contact, and for the skin testing, they literally poke you and purposely put known allergens in to see if what you're allergic to. So they did all down my forearms, across my back. It was brutal. And I was so sick recently that I almost said I'll do it again. But my allergist was like, we can do blood tests and figure out about how many IgEs you have for some of the things that you're allergic to and see where you're at. Because I went through this therapy, this desensitization therapy, where they purposely made a cocktail specifically for me, all the stuff I was allergic to. And they injected it into me. Purposely injected what I was allergic to into my skin at very low concentrations, right? And then slowly increased the concentration. Unfortunately, I only did 6 months of the therapy, and my husband got deployed or got sent for training to California. Even though I brought my stuff with me, we couldn't use it because I was in constant allergic. There was too many grasses there, too much stuff, and I was always inflamed and always taking my allergy medicine, so I couldn't really do the shots because I was so allergic to so many of the things that were there. So needless to say, I was glad that we were only there part-time. 2 years. I was ready to come home. I was like, get me to Louisiana. Get me away from all this nasty stuff. But the good news is I think that because of this desensitization, when they did my last testing, I'm not allergic to thistle anymore, and that was the worst one for me. Ragweed's still bad, and some of the other grasses are still kind of bad. If I would have continued on further along in the course, there's the possibility that I wouldn't be allergic. Why was this beneficial to me, though? Why would purposely giving me something I'm allergic to, how could that help me? Yeah, but if my body's recognizing it, it usually produces what? IgE. But that's because I'm inhaling it. And those cells in that area are programmed by those helper T cells nearby to go IgE. If we inject it, are we going to make IgEs? No, you make IgGs. Did those attach to mast cells or basophils? No. 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 IgGs don't attach to mast cells or basophils, just IgE. So if we can switch the system to go the IgG route and get rid of the allergen before it gets to the IgEs and stop this cycle, right? If we can stop this cycle, get rid of these IgEs, just produce IgG, we can stop this cycle. Okay, so I have recordings to post for you guys as well. I was hoping to get further. Oh, no, wait a minute. My wrong class. Who's packing up on me? Stop! I'm thinking I'm out of time, but I have a whole half an hour. Huh? Yeah, it was. I was like, you know what, y'all? It's the end of the semester, so it really doesn't matter. But when you pack up, right, it's a kind of trigger to the instructor that it's almost time to get out of here because you probably have to run across the campus. If you have to leave early, like, warn me ahead of time. Warn your professor ahead of time. And I'm like, we understand there are things that happen, right? But warn us so that we're not doing like I'm doing, going, I'm out of time, and then I go, no, I'm not out of time. My last class ended at 15:15 the hour. You guys ended 15:15 the hour. I was like, God, man, I was like, I knew I should get through more of this lecture than, don't do that. This one, don't worry about. Don't do that to your poor professors. Okay, this one don't worry about. I haven't investigated on whether they're actually still using this. It was in the textbook at one time, and I don't like to talk about stuff that, yeah, that I don't have knowledge about.
I heard that you're supposed to only take that 5 days because it's causing constriction and messing with the nerve right. So is that like IgE or IgG?
No. Flonase is a drug, and it's actually multiple things. It affects multiple things. And yeah, you should try to limit how much you take it if you can. During the season when the stuff that me and my son are really allergic to, if we don't take it every day, then it doesn't work for us. Because again, it's a preventative, right? So you're taking stuff to, when you inhale, it's going to prevent it. So if we don't have it already in our system, it's not going to help us. You know, and for some individuals, yeah, we got to take it daily. And see, that's the thing is that even me, when my doctor first prescribed it to me, I wasn't using it properly. I was taking it after, you know, and it will help some after, but your best benefit is to have it already in your system. And it's one of those ones where if you don't have to, don't take it, because it can cause other problems because of the type of drugs. I mean, one of them is a steroid. One of them is a steroid, yeah. So, I mean, it has damaging effects. But for some of us, you know, that's one of the few things that works that we can actually breathe through our nose. So the next one is cytotoxic. And if you guys look at our table that we went over last time, are there antibodies involved in these reactions? Yes, yes, right? So again, right, you've got to be exposed to usually the antigen, right, or the allergen, right? In this case, we still refer to them as antigens. And you've got to produce antibodies. And that's why you're going to end up with a reaction, right? Is these antibodies attacking, in this case, it's cytotoxic cells. And we really discovered this hypersensitivity when we started doing blood transfusions and we started having moms having problems with hemolytic disease of the newborn. Because in this case, antibodies are attacking red blood cells because they're recognizing those red blood cells as foreign. So complement fixing antibodies, right? So we know antibodies can react with complement, and complement can activate via the classical pathway, right, with antibodies, and produce that MET complex, right, the membrane attack complex, which you can insert into membranes. So what two classes of antibodies can do that? Activate complement. There's just two. There are two main ones inside our body. IgG and IgM, right? Both IgM and IgG can activate complement via the classical pathway and cause cell lysis via the membrane attack complex. The other is antibody-dependent cellular cytotoxicity. Right, that's an antibody outcome for just one class of antibody. What class of antibody? Which one out of those two? IgG. IgG will bind to these red blood cells, right? And another lymphocyte will come along and kill that cell because it's coating it in IgG. What lymphocyte does that? The natural killer. Right? It naturally kills. That process where you've got a cell coated in IgG and a natural killer cell comes along and kills it is referred to as antibody dependent, right? That natural killer cell is killing that cell because the antibody is coating it. The antibody is recognizing it as foreign. So examples of type 2 hypersensitivities are transfusion reactions and hemolytic disease of the newborn. And this is where antibodies are attacking red blood cells, right? Complement is getting activated and those cells are being destroyed. And or with IgG, natural killer cells are causing the death of that cell. So the major antigens that create problems. And by the way, Tyler recognized that I didn't fill out my second Punnett square correct last time. I don't know if you guys noticed that. I didn't have my things come down correctly from last time when I was talking about sickle cell. But I promised to fill in better Punnett squares today, and actually I'll do it on the computer. we'll just do another slide and insert a table. What do we need? Punnett squares. Yeah, but I need the other squares too. So it would be 6 by 6 maybe. Would that be enough? Let's see. Let's just do that. No, that's too many. I need 6 rows, but only 3 columns. Yeah, that's okay. We won't use the whole thing. So say, right, in the case of my family, my dad was O, right? That means, and this default sucks. There we go. Let's make it so I can see it. He has O-type blood, which means that that gene that he gave me, that DNA that he gave me, does not have any of the antigens that we refer to as A and B. So both of his alleles that he got from his parents, both of his copies of DNA were O's, which means he doesn't make the A or B antigen. My mom, on the other hand, I think is B blood type. She could be, she definitely has at least one allele of B, right? But I know from my blood type, she is not what we call homozygous. She's heterozygous. She has one of her alleles is naked. That's what the O kind of stands for. She doesn't, one of her alleles wouldn't produce any antigens on the surface of her red blood cells. But her other allele that she got from one of her parents does produce what we call the B antigen on the surface of her red blood cells. The reason why I know that she is heterozygous is because I am the same blood type as my dad. So let me fill in my Punnett square correctly this time, right? So I would do my B's across, my O's across, my O's across, and then fill in with all of my dad's O's. Oops. Oh, Erica. Keep hitting the wrong button. There we go. Okay, so these are the possibilities for my parents' children, right? They can have O blood type just like my dad, like I do, right? Or they could have the B blood type just like my mom, but they're going to be just like my mom in that they're going to be heterozygous. Make sense? Still with me? Okay, so sometimes we used to use this for paternity, right? So actually, I'm glad I made too many Punnett square options because then I can just keep writing. So imagine this is actually me and my ex-husband as well, right? Because I'm O and he's B. So my son has to be B or O, right? Those are the only possibilities. Still with me? Could he have A blood type? No, because where'd the A come from? Neither one of us have A. The kid cannot have A. So this, they used to do these sometimes in schools, right? They would do it as an activity, and they stopped doing it because then people found out that, so imagine you're a kid, right? And you know your blood type is A. And you go home and you ask your parents, what are your blood type? Both your parents say O. That's not your mama and daddy, right? Because where the hell did that A come from? Not them. They don't have it. But there are some blood types where, we really couldn't eliminate like that, right? So say if you're heterozygous for A, you're AO, right? And your spouse is BO, guess what your possibilities are? You can have a kid that's AB. You can have a kid that's B, but heterozygous. Why do I keep hitting the wrong thing? You could have a kid that's AO, right, heterozygous, but they're going to be A blood type. Or you could have an O kid. Right? All the possibilities are possible if the two parents are that. Right? So in that case, right, you couldn't as a parent say, oh, that's not my kid either. Right? You've got all the possibilities there for the most part, right? Unless they're, homozygous for A. Where'd the other A come from? Does this make sense to you guys? Interesting thing when it comes to the ABO blood group is that, again, when are you going to produce an antibody? When you see an antigen. So I have O-type blood, right? Am I ever going to see A or B blood? I shouldn't unless some idiot screws up, right, and gives me the wrong type in the hospital. Otherwise, I shouldn't be exposed to positive or negative, or, excuse me, A or B blood. Positive is a different thing, right? But the weird thing is, and probably for protection, there are antigens out there that bacteria have that are similar to our A and B antigens we have on the surface of our red blood cells. The good news is, though, when you're exposed to them, if you have that antigen, remember you're not going to make antibodies against yourself. So you're not going to produce antibodies against yourself. And hopefully you'll produce antibodies against other epitopes on that bacteria. But for individuals like myself, so I've been exposed to bacteria my whole life, and I've run into antigens that are similar to the A and B type. And since I don't have A or B, did I make those antibodies? Yeah, and they probably eliminated that bacteria. They're of the class of IgM, though, right? They're not. We don't tend to make IgG. We don't do that class switch. So what's the good news with IgM as far as being a mother? It doesn't cross the placenta, right? So you don't have to match your baby on the ABO blood group, right? That match doesn't have to happen. But you can't give anybody, right, someone else's blood that contains antigens you don't have, because chances are you have antibodies against those antigens. So in the case for me, you can only give me O-type blood. You've got to give me naked red blood cells from someone else. You give me A, I'm going to attack it. I've probably got antibodies of the IgM class that are going to attack it. They're going to activate complement. We're going to lyse those red blood cells. And then the whole reason why you gave them to me to save my butt is eliminated, and you just created another problem on top of it. Right? You can't give me B either. And oh my God, don't give me AB. It's like double whammy, right? Not a good idea. So the general rule, right, and I tell my A&P students this all the time, if you understand immunology, you understand, do not give anybody anything they don't already have. Don't give them an antigen they don't already have. So who's the luckiest one among us as far as blood typing goes? Because they've got all the antigens. AB. Are commonly referred to as the universal recipients, right? You can give them A, you can give them B, you can give them A and B and O. You can give everything to them, right? Because they don't have what? Antibodies, because they have the antigen. Me, on the other hand, yeah, no antigens, right? Better not give me any. I'm going to have a reaction. So there's a really cool, the Nobel Prize website that I sent you guys to when you were reading those pages and had the cute little cartoons with the cells and the antibodies and stuff. They have an excellent blood typing game, right, to help you with blood typing and learning who you can give what. But as I said, if you remember the general rule, right, don't give anybody an antigen they don't already have, you won't screw up. Right? You won't screw up. Make sense? And again, this is inherited. So that's when we run into the problem with hemolytic disease of the newborn. The antigen for that is referred to as the RH antigen. It was discovered in rhesus monkeys. Sometimes it's also given the letter designation D. Although I have to say I've not seen that very often. When someone asks you your blood type, when you say positive or negative, you're referring to that specific antigen, the RH antigen. And that's why those are the major ones, right? The ABO blood group, what you are, what antigens you have or don't have in my case, in that blood group of antigens, or the RH. So we're actually talking about two separate antigen things when we give our blood type, right? Your positive refers to whether you have RH or not, right? you're positive, you have it, I have it. If you're negative, you don't. Right? So in this case, it's good news if you're positive, right? Because it means you have the antigen. So can we give it to you? Can we give you the antigen if you already have it? Yeah. Because you're not going to make antibodies against yourself, right? If you have the antigen, right, and we give it to you, you're not going to make antibodies against it or have antibodies against it because that's what you are. If you don't have it and we give it to you, will you make antibodies? Yes. And that's the difference with this one, though, is that there aren't any bacteria or other pathogens out there that make an antigen similar to this Rh antigen. You really only have to be exposed to positive blood that contains this antigen to make an antibody. The problem is the class of antibody we make against this antigen. It is Ig. All right. Why do moms have to worry about it? It's IgG. Because what does IgG do as it relates to a baby and its mama? It crosses the placenta. So the only ones who have to worry about this, right, are Rh negative moms. Because if they get exposed to positive blood, they could produce the antibodies of the class IgG that can cross the placenta and attack that baby's red blood cells. And that baby's own complement system is going to lyse its own red blood cells and maybe even its own natural killer cells. Because the baby does not recognize these IgGs as being bad, right? Mommy gives them to me all the time. Why are these ones bad as opposed to the other ones she's been giving me, right? All throughout pregnancy. So we try to prevent this from happening. We try to prevent moms who are negative from producing antibodies against the Rh antigen. So how do we do this? We prevent exposure to positive blood. So if you are negative, you certainly don't want some idiot to screw up with a blood transfusion and give you positive blood, right? Because you're going to make antibodies against it. Now, the very first time you probably won't have a reaction right away, because remember, 17 to 14 days to make those antibodies. Those red blood cells, hopefully enough of them, right, depending, you might end up with a reaction later on. The worst is now if you decide to be a mom, right, it takes two, right, 2 sets of chromosomes, one from mom and one from dad. So even though mom is negative, dad could have what?
Positive.
Positive. That's how you end up with a positive baby, right? So you got a positive baby. If you have antibodies, they cross the placenta, they attack your baby. So one way is getting exposed to positive blood, right? Some idiot giving you the wrong blood in a transfusion. What's another way you could be exposed? How are you going to get exposed to positive blood? Well, that placenta is attached to you, right? Does your blood mix with your baby's blood? No, it's separated. That's the whole purpose of the placenta, right? Stuff diffuses through that zone, right? From mom's blood to baby's blood, right? There is that barrier in there. Is there a possibility that could get damaged and you could have mixing? Yes, right? When severe trauma happens to the placenta, you could have that mixing happening. The other thing is when the baby leaves, it's fine, right? If he survived, mom's negative, but baby's positive, and mom didn't get exposed, she didn't produce antibodies all throughout pregnancy or previous to pregnancy, all's good, baby leaves, right? Then placenta leaves. When that detachment happens, baby's blood could get into mommy's blood. It's a huge wound, right? Placenta's about this big. It's enormous. I've seen a real one. I wanted to keep mine, but I didn't think about it at the time. My professor at the University of Massachusetts at Amherst preserved his grandbaby's placenta and would bring it into lab and show everybody. It's great birth control when you're in college. You're like, oh yeah, I'm good. That's why I was like, I need it for my students. Right? Yeah, it's huge. It's huge. It has a big job to do, right? Exchange nutrients for the baby and waste for the baby when it's in utero. So one of the things that we do for negative moms is that in the birthing process in labor, they're going to give Rogaine. They're going to give passive immunity antibodies against those red blood cells. So any of the baby's red blood cells that gets in, we're going to eliminate them before hopefully mom recognizes them and mounts an active immune response. Unfortunately for some people, for some reason, that may not work, or other exposures they may have had, they end up producing these antibodies. And so therefore having, children, so if you want to know about the agony of that, talk to Dr. LeBlanc, because he has 3 beautiful children, and his wife is Rh negative, and he's probably homozygous positive because all of their children have been positive. And his wife, for her last pregnancy, had to go for shots all throughout her pregnancy to not have antibodies that she's produced to attack her baby. So it can be a real pain. But again, we know about this. We can treat. We can prevent it from killing the baby, right, and having miscarriages because of not matching on this one antigen. Now, is this the only antigens found on your red blood cells? No, these are just the major players. So if you donate blood and you happen to have a really cool, unique blood type, you'll actually get a special card. If you donate blood, you will always get a card from them that has your blood type on it. So that's one way, if you don't know your blood type, of finding it out. And you can help up to 5 other people, right? Because they're going to pack your red blood cells. They're going to take your antibodies. They're going to take your plasma. They split it up 5 different ways, right? And can save 5 different people with just one donation. So if you have the ability to do so, do so. You have the chance to help many people. If you're O positive like myself, they'll keep sending you flyers in the mail, trying to encourage you on free stuff they'll give you to donate, because I'm considered, universal donor as far as O goes. They can give my blood to anybody. The positive does obviously limit me to just positive people. Anyone who is negative can only get negative blood. O negative is the most sought after, right? Because it doesn't contain any of the antigens. We can give it to anybody. You don't have to worry about what antibodies I have, because again, they're going to filter out your red blood cells, and they're giving what's called packed cells, and they don't give your plasma, your serum, to other people. They filter that out. So I did talk too long. So definitely read over that handout as it applies, and I'm going to post an old lecture for you guys for type 2 and type 4. I think, did I talk about type 4 as far as TB tests to some of you guys that are going into health profession, right? Don't be surprised if you end up with a positive TB, if they keep giving you the TB test year after year or every 6 months. It could happen. Don't freak out, right? Just means your immune system's recognizing it. Doesn't mean you have the disease, right? Once positive, always positive. Once your immune system recognizes it, that's it. All right.